Proteins or enzymes are incredibly important molecules and carry out specific function inside of your body. They're activated, or in specific cases deactivated, by attaching to a specific molecule. Each enzyme has a specific shape that corresponds with said molecule, and can only be activated by molecules that have a shape similar to the one in the enzyme. Enzymes are made by translating DNA onto RNA strands. These are called mRNA. Your mRNA then leaves from the nucleus to the ribosome, which is an organelle in the cell. The mRNA is then read for its codons. Each codon is basically 3 bases, and each codon reads for 1 amino acid. So just remember, RNA is read in 3s. Think of it as building blocks. Each amino acid is a building block and will eventually make up a protein. DNA replication doesn't always happen cleanly. In fact, mutations happen all the time, but it's a mistake in the process. These can either A. totally mess you up, or B. leave your enzymes functioning totally normally. A mistake in your DNA can possibly make it so that one amino acid is used instead of the correct one, or completely mess everything up entirely. This is important because the shape of the protein is determined by the amino acid makeup, and the shape determines what it binds with. If it is deformed, then it can't do its intended purpose, and this can either be bad or good. First, we have point mutations, which are substitutions of one DNA base. These can be silent mutations. If you look at this codon to amino acid chart, which you will have on the AP exam, you can see that multiple combinations of RNA bases will code for the same amino acid. If you get super duper lucky and manage to get one of these set combinations, then congratulations! It's just a normal protein. Missense mutations are when you aren't super duper lucky and a different amino acid is substituted in instead of the original. What this results in is a different shape of the protein. However, this does not mean it's always negative. In fact, it's entirely up to chance if it either makes the protein function worse, because in some cases it can even help the protein function better. This is how evolution happens, because a good mutation causes a better trait. Nonsense mutations are when a stop codon appears instead. Stop codons signal to stop building the protein, so when this happens early on in the sequence, this means that the protein's building is basically cut short. This is disastrous because it can make the protein completely non-functional. Next, we have frame shift mutations, which insert or delete DNA bases. These are, compared to point mutations, way worse. RNA is still read in 3s, meaning that if you insert or delete one nucleotide, everything after that nucleotide will be one position off. This means that all the codons are off and can lead to multiple amino acids being different and completely altering the function of the enzyme. If deletions or insertions happen in a multiple of 3, then there's a possibility that it won't have as bad an effect. However, there can be way bigger mutations in the form of chromosomal mutations. Chromosomes are a bunch of DNA packed together into 2 very dense and much larger pieces. Every single cell has these chromosomes, and during cell replication, this replication process can often get mixed up. There are single chromosome mutations, which only affect one chromosome. Duplication is when a specific portion of DNA in a chromosome is duplicated, leading to potential overproduction of the enzymes that portion of DNA makes. This can potentially affect function of the cells depending on the enzyme. Inversion is when a specific portion of DNA is flipped over in orientation inside the chromosome, so it's not the whole chromosome. This can affect the genes inside of the flipped regions. Deletion is when a portion is deleted from the chromosome, and depending on severity, can be incredibly dangerous if the portion includes important information. Translocation is an interchromosome mutation because it affects multiple chromosomes. Translocation is when a part of chromosome breaks off and attaches onto another chromosome. There can also be issues with numbers of chromosomes. Humans are supposed to have 46 chromosomes, and disorders like Down syndrome can be caused by wrong numbers of chromosomes in each cell. During the division of sex cells or meiosis, 2 cells can divide but with uneven numbers of chromosomes, leaving one with too many and one with too little. This is called non-disjunction. So, what have you learned today? Point mutations are a singular substitution of a DNA base. Silent mutations are when point mutations don't change anything. Missense mutations are when point mutations changes one amino acid. Nonsense mutations are when point mutations code to stop early. Frame shift mutations shift the frame of reading codons, leading to everything after the mutation possibly not being correct. Chromosomal duplications are when a portion of DNA is duplicated. Chromosomal inversions are when a portion of DNA is inverted. Chromosomal deletions are when a portion of DNA is deleted. Chromosomal translocation is when part of one chromosome breaks off and attaches to another. Non-disjunction is when cells don't split DNA properly and cause wrong numbers of chromosomes inside the cell. Thank you for watching. If you have any feedback, please leave it in the comments below. And thank you everyone for watching.